Hi guys, hope you all are well. Welcome to my channel Sutapa's Biology. I am Sutapa Datta. My today's topic is continuation of enzyme. In my last videos, I have told you about the structure of the enzyme, about the nature, about the classification and also the mechanism of enzyme action. The two models of enzyme action. Now, after that, it's the most important part for the competitive exams, that's the enzyme inhibition. The function of the enzyme sometimes get inhibited. Now, what are the conditions when the functions are inhibited? It's known as enzyme inhibition. There must be some materials or substrates which inhibit the activity of the enzyme, and according to that, these are classified. Number one is there is some irreversible change, irreversible inhibition. Number one, and number two is reversible inhibition. So from the name, it's clear that irreversible inhibition means where inhibition can't be restored. That means not inhibition, the functions can be restored from the inhibitor. And in case of reversible inhibition, the function can be restored. So these are the two categories. In irreversible inhibition, there is the role of temperature mostly. Like due to high temperature, the enzyme get broken or degenerated, or the denatured, protein get denatured, and that denaturation is known as denaturation of protein. This can't be restored as it's the permanent damage. So this one is mostly effective inhibitor. And in case of the reversible inhibition, there it's also very important one, because you will come to know that potassium cyanide action is also included in this. So it's also very important and very harmful also, but sometimes it can be restored. How it can be and how the function is inhibited, that I am discussing now. Okay, so next is reversible inhibition. And in reversible inhibition, there are some kinds like that. Reversible inhibition. Here, number one is that competitive inhibition. Competitive inhibition. You are very much familiar with the term competitive, as always you have to here and there that you have to appear for the competitive exam, you have to perform the competitive exam perfectly and efficiently. So you are very much familiar with this term competitive. Now competitive inhibition, number two is non-competitive inhibition. No competition is there, but that's the most dangerous one. And number three is allosterism. In some book you will get that allosterism is included in other part. Sorry, allosterism is this one. So but in enzyme action also we shall consider this allosterism or allosteric inhibition or allosteric regulation. Better to write allosteric modulation. allosteric modulation. Now about the competitive inhibition. I am rubbing this other things and coming to competitive inhibition. In competitive inhibition, there must be the competitor. Who is the competitor? The inhibitor is the competitor. Whose competitor? The substrate's competitor. ये तुम लोग जानते हो, आप लोग जानते हैं ये वाला जो इनहिबिशन में मैकेनिज्म ऑफ एंजाइम एक्शन में आप लोग पढ़े हैं, जो सबस्ट्रेट एंड एंजाइम इज मोस्टली इंपॉर्टेंट फॉर द रिएक्शन. सबस्ट्रेट भी रहना चाहिए और एंजाइम भी रहना चाहिए. नाउ हियर अनदर मेंबर इज देयर इन बिटवीन देम who will make the confusion or dispute. That is the competitor, and that competitor is actually the inhibitor. So if I draw the model in this way, this is the enzyme. We know from the last video that substrate should be at par with the enzyme. The structure of the substrate should be at par with the enzyme, and this is like that. This should be of this kind. This is the actual substrate, and the inhibitor should be of this kind, like that. This is the inhibitor. So this is enzyme, this is substrate, and this is inhibitor or competitor. Now inhibitor or competitor can occupy the active site of the enzyme like the substrate, as their structure is almost similar with the substrate, and enzyme will not recognize properly that which one is substrate and which one is competitor. So in that case, enzyme can be occupied by the competitor in that way that enzyme's active site will be occupied by the inhibitor in this way. Enzyme will not understand that it's the competitor or inhibitor. Now after that, there is no chemical matching in between the enzyme and the inhibitor. In enzyme and substrate, they have the chemical matching, and so they will go on with the reaction. But in case of the inhibitor, they have no such similarity or matching with the enzyme. So it can't continue the reaction, and then enzyme molecule will be occupied by them and it will be functionless. So there will be no reaction after that, and ultimately the action of the enzyme will be interrupted in this way. Now in this case, we can give the example of the enzyme is succinate dehydrogenase. Succinate dehydrogenase. Its actual substrate is succinate or succinic acid. Succinic acid, and its inhibitor is malonic acid. Malonic acid. So succinate dehydrogenase is a substrate specific enzyme which can act only on the succinic acid, and after getting combined with the malonic acid, then the enzyme will realize that it's not the proper substrate, but that time there is no way to leave that competitor or inhibitor. So naturally the enzyme action will be come to the halt. Now we'll come to the halt, and so here what should do? This can be, it's a reversible one. You know that this one is reversible one. So if we increase the concentration of the substrate in comparison to the competitor, then the reaction will be restored. Like that, suppose there are 100 molecules of enzyme and 100 molecules of enzymes. इतना enzyme है, enzyme molecule है, और substrate is 40 molecule substrate and 60 molecules inhibitor. तो क्या होगा? Naturally, the active site of the enzyme will be occupied more by the inhibitor. ज्यादा से ज्यादा inhibitor molecule है तो inhibitor molecule occupy कर लेंगे enzyme का active site. Now, if we increase the concentration of the substrate, then suppose it will be 80 molecule of substrate and 20 molecule of inhibitor. So naturally, the reaction rate will be restored gradually. Okay, so this is the competitive inhibition, and next we are going to the non-competitive inhibition. नॉन कॉम्पिटिटिव इनहिबिशन भी क्या है? ऐसा कोई कॉम्पिटिटर नहीं है तो in the substrate will not get any competitor who is competing for the action. कोई competitor नहीं है, लेकिन कुछ देर बाद realization होगा जो तब भी enzyme action नहीं हो रहा है. क्यों? What's the reason? So in case of non-competitive inhibition, in case of the non-competitive inhibition, suppose this is the enzyme molecule and this is the substrate. Now, there is no hindrance between them, so substrate is very relaxed that its position is okay and it can easily react with this, but the non-competitive inhibitor will attach with any other place, not in the on the active site. Active site से active site बगैर ये दूसरा कुछ any place it can occupy and it can change the configuration of the active site or change the structure of the active site. How? Suppose this is the enzyme molecule and the non-competitive inhibitor is binding with this here. पीछे से अटैक करते हैं, सामने से सीधे वे में नहीं, पीछे से अटैक करते हैं और now it will change the structure of the enzyme molecule. How? Just the structure of the enzyme molecule will be changed in this manner. Now substrate is present here. The active site is also free, but the substrate will not be able to attach with this for the chemical reaction. When it coming to the site, then observing that seat is broken, chair is broken. अगर chair टूटा हुआ है तो वो कैसे बैठेंगे? There will be no reaction naturally. So there will be no combination of the enzyme and the substrate due to the presence of the non-competitive inhibitor. Okay, this is very important one as the reaction of the cyanide is included in this. Reaction of cyanide, what will happen with the cyanide? Actually, first I am mentioning about the list of the non-competitive inhibitor. The cyanide compounds, the first one, or cyanide salts, and number 2, the salts of heavy metals. Heavy metals like mercury, gold, etc. So, in case of cyanide, the cyanide, why when it's reversible, then what's the problem of the cyanide? Actually, what happens? Cyanide act on the cytochrome oxidase, which is our respiratory enzyme. Cyanide acts on cytochrome oxidase and immediately it choke the respiration. If it choke the respiration, then for a fraction of a second, if there is no respiration, the organism will die. So immediately the organism will die. I think you have heard about it. आप लोग सुने होंगे जो साइनाइड लेने का बाद पोटेशियम साइनाइड की ज्यादा से ज्यादा नाम है इस मामला में. तो पोटेशियम साइनाइड लेने का साथ साथ तुरंत टांग में जैसे पहुंचे ऐसे डेथ हो जाता है. तो क्यों? बिकॉज इट्स एक्टिंग ऑन दिस cytochrome oxidase, which one is essential for the respiration. And if respiration get choked, then but क्या होगा? उसके बाद उसको withdraw करके organism तो died already. So dead person से वो ये निकाल के क्या होगा? कुछ फायदा नहीं है. तो इसको जिस ये तरह से in case of the other things, that is the salts of the mercury and etc. gold, in that case we can remove this one by dialysis, and after dialysis the patient can live normally. But in case of the cyanide, there is no scope to treat the patient by dialysis, so they will immediately die. ये है non-competitive inhibition, and that's all for today. Our next one will be allosteric modulation. Okay, so after, for that you just observe gradually all the videos, and if you have any problem for understanding, you just make comment in the comment box. Bye.